All right, everybody. It's now my pleasure to introduce Louis Bartlett.
Hello everyone, thank you for having me. It's a real pleasure to be back at Emory where I postdoced briefly to be able to speak about some of this work. A large section of which was actually done at Emory as well as at University of Georgia, Exeter during my PhD, and at UC Berkeley, as well as a whole range of funders having supported some of the series of studies I'm going to be talking about today. Mostly which was a product of a fantastic team of collaborators, Berry Broser, Jaap de Rood and Travis Dines here at Emory during 2 of their tenures here, and Jaap is obviously still around. Keith Delaplane and Jennifer Berry, who are at UGA, my colleagues there, and then Mike Boots now at UC Berkeley. So I work principally on infectious disease in honeybees, and honeybees are fairly important, especially with changing agriculture. As we continue to intensify the agricultural production in the US, they're likely to remain quite crucial to propping up our agricultural activities and our crop productivity. However, this demand is also in some ways the cause of one of their demises. We've seen widespread difficulty in meeting pollination demand with honeybees across the US sector. This is due to facets of the agricultural monocrop intense system of farming we've developed over here, including agrochemical exposure, as demonstrated here, habitat and forage loss, such as the simplification of farmlands into large monocrops. This has mandated a change in beekeeping over the last 5 years towards this migratory, nomadic beekeeping style, with about 2/3 to 3/4 of all managed honeybees in the whole of the contiguous United States being brought to Northern California for the almond orchard pollination, which is a cross-continental kind of mixing event of this crucial livestock. In order to do that, they're packed onto flatbed trucks, such as this one, shipped across the US. Each pallet contains 4 individual honeybee colonies, which will then be placed in an apiary. And this starts to get a sense of the multiple scales at which infectious disease transmission can occur, and has led to changes in their parasite and pathogen dynamics as beekeeping has changed in reflection of the transformation of the US agricultural system. In particular, these kind of events where many millions of individuals are brought into close contact under a stressful ecological situation and then dispersed back across the continent, potentially having exchanged various parasites and pathogens. And that's really where a lot of my focus for the last 5 years in terms of research is concentrated on. So we know that different styles of beekeeping influences the infectious diseases that we observe in them, which we showed in this paper, where we took honeybee populations from 3 kind of distinct beekeeping styles. Feral bees caught from the Okefenokee swamp here in Georgia, which has some fantastic stories. Asked me about my favorite snake and spider stick. It's our most important laboratory tool. Traditional kind of low-intensity beekeeping that reflects the way we've been keeping bees for many thousands of years, and then this modern industrial migratory high-intensity beekeeping. We took bees from these environments, brought them into a common garden sort of experiment, let them equilibrate for a while. Many generations of bees were born and died during this time, and then measured their viruses. And we found a couple of crucial patterns, but the one I really want to focus on today was a consistently higher degree of viral titer in the bees that were sourced from this industrial, novel, migratory, high-intensity management. And this was universal when compared to the low-intensity bees in orange here, even after a full year having been brought into a common garden. So we know that the dispersal of these bees back into landscapes across the US carries with it a tendency towards higher viral titers, which is crucial for a whole range of conservation reasons, as well as the management of the honeybee system. There's lots of explanatory possibilities underpinning this pattern that we observed here. One of them in particular, when we're focusing on viruses, is to do with this ectoparasite, the Varroa mite. This is essentially a tick of honeybees. It's an acarid mite that jumped from a different honeybee species in Southeast Asia about 100 years ago, arrived in the US in the early 80s in about 1984 via Florida. And since its jump into Apis mellifera, the western honeybee, it's been closely associated with increased pathogen and in particular viral burdens in the bee. It's taken us some time to narrow down between kind of different explanatory hypotheses how Varroa interact with either the honeybee immune system via mechanical non-propagative vectoring of various viruses to understand this change from an ancestral case of honeybees having had broadly low levels of viral pathology to it now being the #1 driver of honeybee losses in the US, with some work that's now in revision kind of telling the story of how we've settled on this proof that ancestrally directly transmitted bee viruses have switched towards a vectoring life history, essentially. And so Varroa control remains a really important, crucial part of beekeeping. And we know that Varroa are currently evolving as a vector to our control measures in the same fashion as we've seen happen for things like mosquitoes. We've had some good talks on that. Just over the last couple of years, we've started to see the characterization of the physiological and molecular biological mechanisms of resistance in this species. So we know there's standing genetic variation that Varroa can evolve using in response to different aspects of beekeeping. And one thing we've been wondering about is whether or not parts of the difference we see in beekeeping losses across the sector are based on specialization of this Varroa mite to different ways of beekeeping. So following some of our viral work, we again returned to this variety of beekeeping origin points. And rather than taking bees, we specifically took Varroa, the vector, and inoculated colonies with them to try and assess if there's differential virulence, differential population growth in this crucial mite. Some of this earliest work was done by my PhD kind of cohort compatriot, Charis Dines, who did see some differences in virulence and population growth, but they weren't consistent. So he has two graphs here that I think really start to show the importance of scales in this system. We're on the left, the mites from the high intensity, highly managed background grew to the largest population sizes. So this sticky board measure is a measure of the absolute size of the parasite population in any given colony. But this alcohol wash method is our per capita measure. And so while this one represents the absolute size of the parasite that we observe in any given honeybee colony, this one, this per capita parasitism rate, is where virulence manifests. So we see this kind of puzzling mismatch between the ability of the parasite to grow to a large population and how much it's capable of suppressing the honeybee population, leading to the worst cases of it being kind of virulent. At the same time, Charis also showed that the ability of honeybees to differentiate their home hive and the degree of drift, the movement of honeybees between colonies within an apiary, determined their parasite rates and also their survival. This plays into a wider discussion we've been having in the kind of honeybee health parasite intersection of this idea of what we call mite bomb colonies that are capable of growing very large, with large parasite populations, and then at some point collapse with active dispersal of those parasites into neighboring colonies. And that's been tentatively suggested as one of the major drivers of honeybee losses at the apiary level. As these large colonies grow, pop, the Varroa disperse out into neighboring colonies, and that kind of wreaks havoc. So we set out to test this with some subsequent experiments where we did a similar set of assays to what Charis had done. But rather than just looking at inoculating whole apiaries, we inoculated subsets of colonies and then treated their neighboring colonies, their kind of compatriots within any given farm, apiary, herd, however you want to call it. And then looked to see how the growth of Varroa, based on this genetically varying kind of a range of stocks that we had access to, how that manifested in terms of impacts on virulence or colony loss, based on looking at different scales in this intercolony transmission. And this is what I'm mostly focusing on today, because it's a kind of puzzling story, but I think kind of exciting. So what we observed when we did this subset inoculation and focused on the health of exposed colonies who weren't directly inoculated, but were placed spatially adjacent to inoculated colonies, was at the apiary level, which is where multiple colonies sit kind of close to one another. That's the kind of beekeeping unit of a herd. We saw that the inoculated colonies defined broadly through a significant positive correlation the mite growth, the mite population size in the whole apiary. So our measure of how fast mites were growing in a given inoculated colony defined the whole population, the apiary. However, this wasn't a site-based effect, as during the spring-summer when population growth is occurring, this coefficient didn't correlate with adjacent colonies. So it was purely the inoculated colonies that were driving up the entire patch-level parasite population. When bees enter their critical overwintering, i.e. hibernatory period, these colonies shrink to pretty defined sizes, leading to a much tighter relationship than we see during the summer between mite population size in the colony and per capita parasitism. which is where virulence manifests. So bees go through this critical overwintering period where they shrink during the fall, and these bees are extremely sensitive to how healthy they are in order to allow them to overwinter. What we essentially saw was support for this mite bomb-based hypothesis, where large inoculated colonies were capable of growing large numbers of parasites that then collapsed, dispersed into their neighboring colonies during this critical going into winter period, and led to the death of neighboring colonies without themselves actually necessarily reaching particularly high per capita parasitism rates. So Varroas show variation in their growth rate, and this leads to higher degrees of mortality when colonies are placed adjacent to colonies which contain these fast-growing Varroa strains. And trying to disentangle what this means in terms of measuring their virulence, both on individual honeybees, but also at the colony and apiary level, is something we're really trying to integrate with both beekeeper practitioning work, but also to better understand how spatial scale does influence parasite dynamics in this system. I'm also just going to take a minute to talk about some of the more interesting kind of scale-based influences on this parasite virulence system. And I am constantly reminded by some of my colleagues who aren't as parasite infectious disease focused as me that the other side of virulence is obviously the host factors, and there's multiple scales at place in honeybees here that interact with Varroa growth ability. And one of these that I'm going to focus on just very briefly is genetic diversity. This is an aspect of colony health that's intra-colony. It's about the relatedness of individuals in a colony. And we know that the degree of queen promiscuity, i.e. the genetic heterogeneity in a colony, leads to increased or lower parasitism based on how many different males a queen is able to mate with during her mating flight, whereby they represent diverse subset of sperm that leads to a diverse colony. Now, we don't know initially, prior to some recent work, whether this was about hunting for rare resistance alleles. And so we had a master's student artificially inseminate a bunch of honeybee queens, measure their survival, and found principally that the higher the genetic diversity within a colony, the lower the degree of parasitism. So we do have kind of an intra-colony scale effect here of genetic diversity, determining Varroa growth rate and then subsequent survival. So actions like this of policing parasite levels. I'm going to skip through to the end now because there was one other exciting experiment, but it's not really relevant to you all. It's about pesticide exposure, but do ask me about it. And I'd be delighted to take questions, but I'd also like to announce that as of 3 weeks from today, I'm being promoted at faculty at University of Georgia, and I would be delighted to recruit master's students in particular who want to work on this kind of agro-infectious disease virulence evolution system. I'd be happy to take questions.
Thank you so much, Lewis. I think we have time for about 2 questions. Or any master's students looking for jobs?
Hi, I wanted to ask, there are like interventions that like beekeepers can do if they have these mite levels. In these cases that you have these like highly like parasitized colonies, is it kind of like a runaway effect? Is there any like, is intervention helpful in these scenarios for that colony or at the whole apiary level?
Yeah, I think that's a really good question, Alison. I actually had an argument on that exact topic with the reviewer of this work recently, so excuse me if I get a little lively during this. I think the crucial thing here is this disentangling based on the per capita versus absolute size of the parasite population in any given colony. Where for a beekeeper, they're going in and they typically measure the per capita parasitism rate because that's what determines the health and kind of vitality of the colony. But in any given apiary, the largest Varroa populations are actually going to be in the largest colonies because that's the principal determinant of size. And so based on this work, at least in Georgia, we've shifted towards recommending that beekeepers treat for this parasite at the apiary level rather than at the per colony level, but that's something that has apparently not been widely accepted by every single one of my colleagues. So I think there's still some nuance There's still some nuance to be worked out in how understanding this difference between absolute and per capita parasitism in this system relates to actionable recommendations for pesticide use, especially when we know that they're evolving resistance to it, and there's kind of the IPM aspects that we have to be mindful of as well.
All right, thank you so much.
All right, thank you, everyone.